Okay, so a very good evening, all of you. Uh, so today we'll be practicing some questions which are related to the week 2 course content, which is basically differential gene expression. Okay, so before we actually start the class, I'll just give you a brief about what is differential gene expression. Okay, so basically all the cells of the body, most of the cells of the body, have the same genomic content. Okay. So, but the genes which are expressed in the lungs, they are not expressed in the eyes. The genes which are expressed in the eyes are not expressed in the lungs. But does it mean that eye specific genes are not present in the lungs? No. Eye specific genes are also present in the lungs area. Then how come that the gene expression is so tissue and cell specific? What determines the cell and tissue specific expression of genes? Can anybody tell me? What is the reason that although all the cells of the body have the same genomic content, still some genes are expressed in certain parts of the body and some genes are not expressed? So what can be the reason behind it? Anybody? Okay, no one? So it is because of the differential gene expression. This is because of differential gene expression. That means although all the genes are present in all of the cells, but the expression of those genes is cell, tissue, and developmental stage specific. What I'm saying? The expression of genes. Hmm, expression of genes is cell, tissue, and developmental stage specific. This means that the genes which are responsible for, let's say, eye lens color, those genes will not be expressed in your lungs or in any other part of the body, and they will be specifically expressed in your eyes. That means although the genes, let's say the gene is X, which codes for the eye lens color, now although this gene is present in all the cells of the body, this gene is also present in your lungs, in your heart, and everywhere, but the expression of this gene only occurs in your eyes. So that is which this is because of the differential expression of genes. That means the expression of genes is cell state specific, tissue specific, and developmental state specific. Okay, so this is the core topic in which we'll be dealing throughout this 2 hours class. So what I'll do is that we'll solve questions, and then with those questions we'll try to cover the topics which are related to those questions, as we always do. Okay, so the first question is, which of the following transcription factors can bind to the enhancers even in the heterochromatic? Strong transcription factors such as OCT4, pioneer transcription factor that is PAX5, potent transcription factor that is SOX2, and pioneer transcription factor that is PBX. So I'll give you 2 minutes to think, and then I'll switch the screen, and then we'll see what you have answered. Okay, I think someone wants to join the meeting. So I'm switching the screen. I hope that you have answered the question. Okay, someone has. Okay. Okay, so two students have answered. Nabandita and Camelia has answered D, and Ajay has answered PAX 7. So let me see the options and then I'll tell you. Okay. Pioneer transcription factor. So all of you have answered it correctly. So the concept is that in the heterochromatin area, okay, so you have your chromatin is organized as euchromatin and heterochromatin, okay. So euchromatin is the part which is actively expressing the uh, transcription. That means euchromatin is the part where transcription takes place. It is the expressed area of the chromatin, whereas heterochromatin is the area where transcription does not takes place, and therefore it is the repressed area of the chromatin. Okay, so all the genes which are expressed in a particular cell or a stage, they belong to the euchromatin area, and all the genes which are uh, not expressed in a particular cell or a developmental stage, they belong to the heterochromatin area. Now, heterochromatin area is actually more condensed than the euchromatin area. So, if any particular gene has to be transcribed in that heterochromatin area, the very first and foremost step is the loosening of the heterochromatin area. If this is the heterochromatin area and some transcription factor has to come and bind to it, then this heterochromatin area has to be loosened up so that there is a place for the transcription factors to come, sit, and bind. Okay, so excuse me. So this is the basic thing. Now, what are the transcription factors that comes and bind? So as if you have gone through the lectures, you must have read or you must have studied that transcription factors, it binds to the promoter regions and it starts the transcription. Now what causes those transcription factors to come and bind? Those transcription factors are activated by another transcription factors. Those another transcription factors are activated by some another transcription factors. So there is actually a kind of a cascade where several transcription factors come and activate each other. But at first there are certain transcription factors which comes for the very first time and it starts the transcription process. It starts the activation process, and those transcription factors are known as pioneer transcription factors. So pioneer transcription factors are those factors which for the very first time comes and binds to the heterochromatin or a repressed area, and it initiates the active process, it initiates the transcription process. Okay, so is it clear? What are pioneer factors? Is it clear to everybody? I hope it is clear.
Yes, ma'am.
Okay, fine. So now PAX 7 and PBX both are pioneer factors, so both the options are correct. Okay, so if there is an heterochromatin area, very first thing that you have to understand is that if you want the expression of any gene which is present in the heterochromatin area, the very first step is the loosening of the chromatin structure so that it provide there is some space for the transcription factors to come and bind. And the very first transcription factor that comes and binds and initiates the transcription process, that kind of transcription factors are known as pioneer transcription factors. For example, PAX5 and PBX7. So the correct answer are both PAX5 and PBX7. Okay, now second question is, neural restrictive silencer elements are expressed in non-neural cells only, expressed in neural cells only, cis-acting elements that restrict gene expression to neurons only, or trans-acting elements that restrict expression to neurons only. I'll give you 2 minutes. Think and then try to answer. After 2 minutes, I will switch the screen. Could you understand the question? After 1 minute, I'll switch the screen. Okay, few more seconds. Fine. Now I'll switch the screen. Let's see what you have answered. Hmm. Okay. So Camelia has answered A and Navanita has answered D. Okay. Hmm. So the correct answer is this. So I guess both of you have answered it incorrectly. Doesn't matter. I'll explain the concept. Okay. So first you have to understand the difference between cis acting elements and trans acting elements. Okay. So what are cis acting and trans active elements? So for a particular gene, you have two copies, you have two alleles. Let's say this is your allele 1 and this is your allele 2. Okay. Now there is uh, a certain factor, let's say A, right? This A is present in 1 and another factor, let's say B. Okay, fine. This B is present on 2. Now this A is a factor or an element which can affect the transcription of only one of the allele in which it is present. That is the first allele, right? And just a minute. And the another factor, which is the B, it can affect the transcription of 2 also and 1 also. So what would be cis and what would be trans? Can anybody tell me which factor can be called as cis and which factor can be called as trans in this case? Anybody? No?
Ma'am, can you repeat the explanation?
So there are two factors A and B. A is present on allele 1 and B is present on allele 2 of the same gene. One gene has two allele. Now this A can affect the transcription of only one of the allele, that is 1. Whereas B can affect the transcription of allele number 2 also and allele number 1 also. So which factor acts as a cis acting element and which factor acts as a trans element? This is the question. Can you tell? Fine, I'll tell you. So, cis acting elements or cis acting factors are the factors which can affect the transcription of only the allele in which they are present, and B or transacting elements which can affect the transcription of the allele in which they are present as well as the adjacent allele. So A is your cis acting factor, whereas B is your trans acting factor, because A can affect the transcription of only one, whereas B can affect the transcription of two in which it is present and one both. Trans. You have cis and trans in biochemistry also, if you remember. So cis means on the same side and trans means on the opposite side. So cis means on the same chromosome, trans means on the different chromosome. So is cis and trans now clear? What is the meaning of cis acting elements and trans acting elements? Cis acting elements can affect the transcription of the gene on the chromosome in which they are present, not the adjacent. But trans acting element can affect the transcription of adjacent chromosome also. Okay, this is the difference. Now the question here is that neural restrictive silencer elements, that means There are three kinds of elements which are actually important for the transcription process. So just to make a background, we are studying about differential gene expression. So when we are studying about differential gene expression, that means same genes which are present in all the cells of the organisms show different gene expression in different cell types. Now why the genes are getting expressed differentially in different cell types is because their expression is restricted because of certain elements, and those elements are your promoters, enhancers, silencers. These are the elements which restrict or promote the expression of genes in different locations. Now, silencer are the elements which restrict the gene expression. Okay. So, silencer means silences. Enhancer means enhances. Promoter means promotes. So, silencers are the elements which restrict the gene expression. This is what silencer is. And silencer is a cis acting element. Always remember, silencers are cis acting elements. Okay, they are not trans acting elements, they are cis acting elements. One thing. Second thing is neural restrictive silencer elements. That means those silencer elements which restrict the expression in neural area only. That means neuron only. So, neural restrictive silencer elements are the cis acting elements that restrict gene expression to neurons only. Is it clear or not? Is it clear to everybody? Just give a thumbs up if it is clear. Yeah. Okay, I think it is clear since I cannot see any response. Anyways, fine, good. So, always remember that the differential gene expression happens because we have enhancers, we have silencers, we have promoters. Okay, so these are the various elements, either cis or trans, which helps in the differential expression of gene. Now, next question is important question. Conditional knockout of a gene by Cre recombinase is possible in which of the following? Now you try to understand this and try to answer. If you are unable to answer, at least try. I'll explain it anyways. 2 minutes to answer this question and then I'll switch the screen. Think and try to answer. Okay. Now I'm switching the screen. Hmm. Let us see what you have answered. Okay. So only Nabandita has answered. Nobody has answered. Why? She has answered C. C means okay. Hmm. Why nobody has answered? Abina, Amina, Ananya, Camellia, Shubhamoy. What do you think should be the answer of this question? How many of you have gone through the lectures? The week 2 lectures? How many of you have gone through them? Have you gone through them? Have you read? Okay. Okay. Good. It's fine if you're unable to answer. Okay. Fine. So, the correct answer is A and B. I think so. Let's see. Yes. The correct answer is A and B. Now, why? Okay. Let me explain you. Now, this, if I don't know whether you are bachelor's or master's students, but if you are thinking of pursuing research or going for PhD, this Cree-Loxp system is a very important topic. Why? Because this will be used if for any of the knockout strategies that you have to do. So if you're not doing like if you're doing mouse related experiments, then you have to use knock in knockouts. Then this Cree-Loxp system is very useful and it is very important, not only for your research per se, but your competitive examination point of view also. This Cree-Loxp system is important. So I would request everybody to go and read about this Cree-Loxp system. But remember that Cree-Loxp system, Cree-Loxp system works on an enzyme which cuts the gene which are between the Loxp sites, and that enzyme is hmm, that enzyme is your Cree recombinase. And whether it will cut or it will invert or what would be the consequence, it depends on the direction that we'll not discuss right now. Just remember that Cree Loxp in this, Cree stands for Cree recombinase. Okay? And Loxp are the sites. So suppose if this is your gene, this is your gene. Uh, I'm sorry for my really extremely bad handwriting. I'm still not used to using this bad writing pad for teaching. Anyways, yeah. So if this is your gene of interest, which has to be knocked out, fine. Then if there is one Loxp site here and one Loxp site here, so this particular gene of interest is removed when this Cree Loxp comes and uh, acts on it. So, if you have to remove a particular gene, then the gene should be flanked by LOXP sites. So, if the gene is flanked and it is removed, it is called as floxed. Why floxed? Because it is flanked because with the LOXP site. So, it is known as flanked with the LOXP sites. So, FL for flanked and LOXP, so it is known as floxed. So the gene has to be floxed in between the LOXP sites in order for the gene to be removed. Okay, so now the question here says that conditional knockout of a gene by Cree recombinase is possible only. So red one is the gene which has to be knocked out and blue is the LOXP site. Now you see here in this case, so this is your one LOXP site and this is your another LOXP site. So not the complete gene, but at least this portion of the gene would be knocked out. Okay, now in this case this portion of the gene would be knocked out because the two LOXP sites are here. The complete gene is in between. Here this gene is will not be knocked out. Why? Because two Loxp sites are here. This portion can be knocked out, but not this portion. Here only one Loxp site is there, so nothing would be locked out. So if you want to knock out a particular gene, then that gene has to be in between the Loxp site. There should be one Loxp site downstream, one Loxp site upstream, and then only your gene of interest can be knocked out. Okay. So this is basically used in conditional knockouts. Okay. Now, can you tell me what is the meaning of conditional knockout? What is the difference between normal knockouts and conditional knockouts? Okay. So, uh, Nabandita and Devnath, sorry, Camelia, do you have any query, or you raised hand earlier because I asked? Uh,
Sorry, ma'am.
Fine.
By
No,
mistake. Sorry, ma'am.
it's fine. Okay. So, do you know the difference between a conditional knockout and a normal knockout? Okay, I'll explain. So, suppose a particular gene has to be knocked out from the whole body, then it's okay. But if I want to knock out a gene specifically from from my spleen cells, let's say, or from my lung cells, or from any particular organ, that means I am giving a condition that yes, I want to knock out this gene, let's say X. I want to knock out this X gene, but I want that this gene should be knocked out only from the spleen area and not from the whole body. So, such kinds of knockouts where you are specifically telling that the gene has to be knocked out from a specific part of the body, that particular kind of knockouts are known as conditional knockouts. Conditional knockout. So whenever we write knockout, in short, we write it like this. Okay. Now, how to ensure that the gene, let's say the name of the gene is X, how this gene X would be knocked out only from the spleen and not from any other area? Because the promoter that you will be using, that would be specific to spleen. So your Cre recombinase will be transcribed only when it gets a promoter which is spleen specific. So Cre recombinase ultimately two things are needed. First thing is that your gene should be in between the Loxp sites, and the second thing is that Cre recombinase should be active. That means Cre recombinase should transcribe. So if you are putting a promoter, if this is your Cre gene, and before the Cre gene you are putting a promoter which is specific to spleen, that means this gene Cre gene would be transcribed only in the spleen area. Then what will happen? Although this Cre gene would be present in whole of the body, the LOXP is present in whole of the body, but because this Cre is getting transcribed only in the spleen area, therefore knockout would happen only in the spleen area. So tissue specific promoter has to be used to determine that in a particular tissue only you want to knock out a gene. So if this particular spleen promoter is replaced by any general promoter, then that gene X would be knocked out from the whole organism. Is it clear? The difference between a conditional knockout and a normal knockout is that for conditional knockout, you are doing the knockout, or you are removing the gene only from a particular area of the body, and for that you are using a tissue specific promoter. Whereas a normal knockout, you are knocking out the gene from the whole body. Fine. Now we'll come to the next question. Hmm. This is the next question. Transposable element used in enhancer trap contains following elements or elements: weak promoter, reporter gene, strong promoter, and transcription factor. 2 minutes to think, and then I'll switch the screen. Could you understand? After 1 minute, I'll switch the screen. Okay, I think there is some question. Just few more seconds and then I'll switch the screen. Okay, I'm switching the screen now. Let's see. Okay. Hmm. Fine. So, hmm. Namandita has answered A and C. Camelia has answered B. Ajay has answered A and B. And okay. So Namandita has answered A and B. Okay, let me see the question again. What was the question? Hmm. Transposable element used in enhancer trap contains following elements. Hmm. It contains a weak promoter, reporter gene, strong promoter, and transcription factor. So the correct answer is it contains a weak promoter and it contains a reporter gene. So let's see how many of you have answered. So, okay, I think everybody has answered it correctly only. So fine. Now, let us see here. I don't remember it was in which lecture. Lecture 5. Hmm. Hmm. So, what do you mean by enhancer trap? Enhancer trap is basically a method to identify the enhancers. Okay, it's a method to identify enhancers. Now, for that, what they are using a weak promoter and a reporter gene. So, let us try to understand this with the help of this diagram cartoon which was already being taught to you by the sir. So you see this is the gene which is regulated by this enhancer. This is the enhancer and this enhancer regulates this gene. So it is a cis regulation or trans regulation. Can anybody tell me? Is it a cis regulation or trans regulation? This enhancer is activating this gene. So is it a cis regulation or a trans regulation?
Cis.
Cis regulation. Good. Why? Because this enhancer and this gene they are present on the same chromosome. If they would have present on a different chromosome and this enhancer regulates the expression of a gene which is present on a different chromosome, then that would be a trans regulation. It is a cis regulation. Now we want to identify enhancer and its activity. So what we'll do is that along with this gene we are attaching a segment of DNA, and this segment of DNA is a transposable element. So, transposable elements are known as jumping genes, which can detach or which can cut themselves from one place and then they can paste themselves in another place, or they can copy themselves from one place and they can paste themselves in another place. So transposable elements are actually very easy to cut or copy from one area and attach to another area. So that's why a transposable element was taken. Now, this transposable element was made in such a way that a reporter gene along with a weak promoter was attached. Now, reporter gene means that if this enhancer will enhance the transcription of this gene, because this particular cassette is also attached, the reporter gene will also be get transcribed. And this reporter gene is either radio lab labeled or it is fluorescently labeled. Mostly you can have GFP, RFP. Nowadays, people mostly use fluorescent labeled reporter genes. So it this reporter gene is fluorescent labeled. So you it gives a color that gives an indication of how much enhancer is regulating the transcription. And this is a weak promoter. Why it is a weak promoter? So that the intrinsic activation of this gene is very minimal. Whatever expression of this gene is happening, it is happening because of this enhancer only, and not because of its own promoter. That is why the promoter has to be a weak promoter. So now what will happen, you are trapping the enhancer in such a way that when this enhancer is enhancing the activity of this gene, along with this gene the reporter gene is also getting transcribed. And since this reporter gene is a fluorescently labeled gene, therefore it gives a color, blue or green color or red color, whatever it is, and then you can um, detect the activity. Fine. And the weak promoter, there is a weak promoter. Weak promoter is because you have to make a cassette, you have to give a promoter, right? But the promoter is weak, so whatever transcription is happening, it is because of the enhancer is enhancing the transcription. Fine. Because see, this is a reporter gene and this is the enhancer. Enhancer will enhance the transcription. Enhancer cannot initiate the transcription. Fine. So for initiating the transcription, there has to be a promoter. But the promoter is weak, so that the amount of transcription is greatly dependent upon the enhancer activity. Okay? Is this concept clear to everybody or not? If it is not clear, I can explain it again. Just tell me, is it clear or not?
Ma'am, can you please repeat again?
Okay. So, um, I'll explain with here only. See, we want to see whether this enhancer has some activity or not. I am saying that this enhancer enhances the activity of this gene. How to believe that this enhancer is enhancing the activity of this gene? What I'm doing is that in between this enhancer and a gene, I am putting a cassette, means a particular DNA sequence, which is a transposable DNA sequence, in which I am putting a reporter gene. So this, if this enhancer is actually enhancing the activity of this gene, it will enhance the activity of this gene also, obviously, because it is in between it. Now this reporter gene is fluorescently labeled. That means when the transcription of this gene will happen, then it will fluoresce and it will give some color that can be detected. Fine. Now, this the activity of enhancer is to enhance the rate of transcription, but not initiating the rate of transcription. That means to initiate transcription, you need a promoter sequence and an initiation codon so that the transcription is initiated. Okay, so that is why for initiating the transcription process of this reporter gene, a promoter is attached. But the point which is important here is that this promoter has to be a weak promoter. Why? Because if this promoter is a strong promoter, promoters can be either a weak promoter or a strong promoter. Strong promoter does not need the activity of enhancer. It can do a good transcription on its own. So if we attach a strong promoter here, then how can we say that the transcription of the reporter gene which is happening, it is happening because of the promoter or because of enhancer? Because the promoter is also strong. That is why the promoter has to be a weak promoter, so that whatever transcription of the reporter gene is happening, it is happening because of enhancer. Sure shot, we can say it. Otherwise, if the promoter is also a strong promoter, then we don't know whether the transcription is happening because of the promoter or because of the enhancer. So, is it clear?
Yes, ma'am.
Okay. Now, let us see the question. What the question was saying. Now, transposable element which is used in enhancer trap contains what? It has to have a weak promoter and a reporter gene. If there is a strong promoter, then we don't know that transcription is happening because of the strong promoter effect or because of the enhancer effect. So therefore the promoter has to be weak. And transcription factor, that is not important. Weak promoter and reporter gene is important. Now, next question is, which of the following is true about the chromosome diminution observed by Boveri? Okay, the phenomenon observed is a result of chromosome condensation and is well conserved throughout the class Platyhelminthes. The phenomena was first observed by Nettie Stevens and it formed the groundwork for genomic equivalence. The phenomenon supports the cell theory proposed by August Weismann. The phenomena was discovered by Christian Wolff in support of the chromosomal theory of inheritance. It supports Weismann's germplasm theory. So you have to tell uh, that chromosome diminution is a phenomena, and which of the following sentences is true about this chromosome diminution which was observed by Boveri. I'll give you 3 minutes. Think, read all the options carefully, and then try to answer this question. It might take a while for you because it's a long question. Think and then try to answer. After 3 minutes, I'll switch the screen. Think of all the options carefully. Are you able to understand the question? Okay. Okay, so then I'm switching the screens. Hmm. Okay, let us see what you have answered. Just a minute. Hmm. Okay. So Namandita has answered E, Camellia has answered E, and Shubhamoy has also answered E. E, which is it supports Weismann germplasm theory. Okay, so I think all of you have answered it correctly. Very good. So, can anybody tell me what is chromosome diminution theory and how it is related to Weismann's germplasm theory? Can anybody tell me? Shubhamoy, can you tell? Can you explain? Think, Camellia, Navannita. Nobody? Okay, fine. Let us go through this. So the correct answer is it supports Weismann germplasm theory. How? We'll see now. So first of all, the concept is genome equivalence. What is the meaning of genome equivalence? Genome equivalence means that most of the cells of our body have same set of chromosome. Why I'm saying most of the cells? Because some cells do not have the nucleus, for example, RBCs. So genome equivalence means that most of the cells of our body have the same set of chromosome. Now, after the genome equivalence theory came the theory of chromosomal theory of inheritance, which said that chromosomes are indeed the important matter, or chromosomes are indeed important for the inheritance from one generation to another. So it was proven by various well-renowned scientists such as Theodore Boveri and Stevens and Wilson. And then came the theory of August Weismann, which is the germplasm theory. And this theory has actually distinguished the germ cells with the somatic cells. Okay, so before coming to the germplasm theory, you should understand what is the theory of chromosome diminution. So, chromosome diminution is actually nothing but chromosomal fragmentation. Okay, so chromosome diminution means chromosome is getting fragmented. Chromosome fragmentation followed by elimination of part of the chromosome during mitosis. Now that means, okay, so chromosome is, as the mitosis is occurring, chromosome is getting fragmented. Now how is it related to the germplasm theory? Germplasm theory actually stated that from a germ cell you have uh, uh, germ cell also comes and other somatic cells also arises. Let us see here. I think from here it would be see, this is the germplasm theory, that a germ cell can give rise to a germ cell and all other somatic cells, and these somatic cells uh, differentiates to all other cells of the body, and this germ cell remains the germ cells, which leads to the continuity. So basically there is a difference between germ cells and somatic cells. So germ cells can give rise to germ cells as well as all other somatic cells. This is what the germplasm theory basically uh, told to us. Now, here we say that chromosome diminution means the fragmentation of the chromosome by subsequent mitotic divisions. So, important point to consider over here is that chromosomes of somatic cells break down while the germ cells chromosome remain intact. Okay, so it was observed through this chromosome diminution phenomena that chromosomes of the somatic cells were breaking down while the cells chromosome remain intact. That means there are two kinds of cells. The behavior of both the cells are different, and it supports the germplasm theory, which states that germ cells can give rise to germ cells. Here it is. Germ cells can give rise to germ cells and maintain the continuity, whereas all other cells arise from the germ cells. So, this this chromosome diminution theory, it kind of inclinates with the germplasm theory, which was given by August Weismann. Fine. Now, the question here was, which of the following is true about the chromosome diminution observed by Boveri? So this chromosome diminution actually it supported the Weismann germplasm theory. Okay, the phenomena is observed as a result of chromosome condensation and is well conserved throughout the class of Platyhelminthes. There is no relation of chromosome condensation here. Here, chromosome is getting fragmented, not condensed. The phenomena was first observed by Nettie Stevens and it formed the groundwork for genomic equivalence. No. The phenomena supports cell theory proposed by August Weismann. It actually supports the germplasm theory. Now, next question is again a big question, but I'm happy that you are able to answer the questions correctly. It means that you have gone through the lectures and you have read it and you are able to understand it. That's appreciable. Okay. The question is, which of the following experiment is likely to generate evidence for genomic equivalence? Transplanting intestinal cell nucleus into an enucleated oocyte, fusing an enucleated oocyte with a diploid oocyte nucleus, artificially fusing two haploid oocytes, fusing a skin cell with an enucleated oocyte, fusion of a haploid neuron with a haploid oocyte, fusion of enucleated sperm with a diploid oocyte, de-differentiating a somatic cell to an embryonic cell and allowing it to develop into a complete organism by transplanting in vivo. So there are many options. I'll give you 3 minutes. Read all the options carefully, try to understand, and then try to answer. Okay, 3 minutes for this question. Read and reread and reread all the options, then only you will be able to answer it. It's a little confusing. Try to answer the question. I want more and more participation in this question. Okay, now we'll see. I'm switching the screen now. Hmm, just a minute. Fine. To see what you have answered. Hmm. Okay. So Navandita has answered G, Ananya has answered G, and Camelia has also answered G. Okay. Let me see the correct option. Which of the following experiment is likely to generate evidence of genomic equivalence? So, first thing is, if you see here, if we try to study evidence for genome equivalence, so the evidence for genome equivalence can be done from somatic nuclear transfer. So, somatic nuclear transfer can be an evidence for genomic equivalence. Okay, so that means we have to see the option which correctly describes the somatic nuclear transfer technique, SCNT. We call it as SCNT. So remember that evidence for genomic equivalence is through somatic nuclear transfer, somatic cell nuclear transfer. Now let us see transplanting intestinal cell nucleus into an enucleated oocyte. So we are taking the nucleus of an intestinal cell, that means we are taking a somatic nucleus and we are putting into an enucleated oocyte. This is what somatic nuclear transfer is. No, see, if you just see here, if you just go through this particular diagram of how somatic nuclear transfer occur, and this was the first evidence of somatic nuclear transfer where a cloned animal, which was Dolly the sheep, was created. So now you see, this is the Scottish blackface strain. This sheep has given the oocyte. So it is the oocyte donor. So from its oocyte, the eggs are removed. So now this oocyte does not have any egg. Therefore, it is known as an enucleated egg. Okay. I mean, does not have any nucleus, so it is known as an enucleated nucleus. So the nucleus from the this particular egg has been removed. This is the egg with no nucleus. Now, nuclear donor, the sheep strain was Finn Dorset strain. From the udder cells, you can see that nucleus was taken. I mean, the cell was taken. So this udder cell was uh, transferred into the enucleated cell, and then with the help of electric current, the enucleated egg and the cell were fused. Now, since the cell has a nucleus and this enucleated egg does not have the nucleus, that means the nucleus in this particular egg is the one which is coming from the somatic cell, udder cell, and then it was grown and it converted into blastocyst. So this is how your somatic cell nuclear transfer has occurred. So, in general, we can say that for a somatic cell nuclear transfer, we need an enucleated egg, enucleated egg. That means an egg or an ovum from which the nucleus has been removed, and a nucleus donor cell, a somatic cell in this case. These are the two important uh, things which are needed for somatic cell nuclear transfer. And somatic cell nuclear transfer is nothing but it is an evidence for genomic equivalence. So now we have to say transplanting intestinal cell nucleus into an enucleated oocyte. It looks correct only. Why? Because see, you have an enucleated oocyte in which the nucleus has been removed, and you have the cell nucleus of a somatic cell. So it, to me, it looks correct. We'll see the correct answer. Then fusing an enucleated oocyte with a diploid oocyte nucleus. No, both are oocyte and oocyte. You do not fuse one oocyte with another oocyte. You fuse one oocyte with another somatic cell. So this is wrong. Artificially fusing two haploid oocytes? No. We are not fusing oocyte and oocyte. We are fusing oocyte and a somatic cell. Wrong. Fusing a skin cell with an enucleated oocyte, this looks correct to me. Why? Because you're taking a skin cell which has a somatic nucleus and an enucleated oocyte, I mean oocyte in which the nucleus has been removed. This looks correct. Fusion of a haploid neuron with a haploid oocyte, no. This is haploid, that means still something is there. No, it has to be enucleated. Incorrect. Fusing of enucleated sperm with a diploid oocyte? No. The oocyte has to be enucleated and the cell has to be a somatic cell, not a germ cell. So it is again wrong. De-differentiating a somatic cell, okay, to an embryonic cell and allowing it to develop into a complete organism by transplanting in vivo. This looks a little controversial. Let us see what is the answer. Yeah. The answer is transplanting intestinal cell nucleus into an enucleated oocyte, fusing a skin cell with an enucleated oocyte, and de-differentiating a somatic cell to an embryonic cell and allowing it to develop into a complete organism by transplanting in vivo. So these are the three options which are correct. So you have chosen one of the correct options, and yeah, you have chosen one of the correct options, but these two options are also correct. So, could you understand the basic concept of of somatic cell nuclear transfer? Somatic cell nuclear transfer precisely involves an enucleated oocyte. That means an oocyte from which its nucleus has been removed, along with a somatic cell which will provide the nucleus. So the nuclear donor or the nucleus donor is the somatic cell, and your oocyte is enucleated. So in these options, along with your G option, you can see this is also enucleated oocyte, here also enucleated oocyte. Here the nuclear donor is a intestinal cell, and here the nuclear donor is a skin cell. So therefore A, D and G, all three are your correct options. Fine. Now let us move on to another question. Oh god, it's such a big question. Fine. Let's see. Which of the following statements is or are true regarding gene and their expression? Cytoplasmic processing involves the addition of 7-methyl guanine cap at the 5' end of the pre-mRNA transcript. Cytoplasmic processing involves the addition of a 5-methyl guanine cap at the 5' end of the pre-mRNA transcript. The poly A tail is followed by the 3' UTR in the processed transcript. The promoter is always found at the 5' end of the gene and comprises of an 80-rich sequence. Gene silencing involves methylation of H2A, H3, H4. The H2A, H4 dimer forms a tetramer by binding to H2B, H3 dimer. This tetramer associates with another tetramer to form the core octamer. Cytosine methylation is well conserved mechanism for gene expression, and TF2H mediated phosphorylation of tyrosine residues is required for transcription initiation. So there are so many 1, 2, 3, 4, 5, 6, 7, 8 options are there. Okay. So I'll give you 3 minutes. If you need more time, you can take more time. We have ample amount of time, no worries. But read all the options carefully and try to answer the question. Take your time, but try to understand all the options. Okay, I'll give you 3 minutes and then I'll switch the screen. Read all the options very carefully and then only answer. Okay, I'm switching the screen now. Shall I switch the screen? Okay, let us see what you have answered. Okay. Hmm. So, Camellia has answered G. Namandita has answered D and F. And okay, you have answered a lot. Okay, that is different. C, D, H, and G. Okay. So let us see what are the correct answers here. Fine. Which of the following statement is true regarding their expression? So cytoplasmic processing involves the addition of 7-methyl guanosine cap at the 5' end of the pre-mRNA transcript. True or false? False, because the addition of 7-methyl guanosine cap occurs at the nuclear level. It is not a cytoplasmic processing, first of all. It is a nuclear processing. This is incorrect. This is obviously incorrect because it is 5-methyl guanosine cap. No, it is 7-methyl guanosine cap. It is incorrect. Now, the poly A tail is followed by the 3 UTR process in the process transcript. No, because the poly A tail is present. Poly A, if you can see here, I have kept the diagram for you to show. See, this is the poly A site and this is the 3' UTR site. So you can see that in the 3' UTR site, you have translation termination codons. Okay, that you have poly A site and transcription termination sequence. That means the statement here says that poly A tail is followed by the 3' UTR in the process transcript. It is also wrong. Then the promoter is always found at the 5' end and it comprises of an 88 sequence. It is wrong. Because why it is wrong? Can anybody tell me? Can anybody tell me? Okay, so it is a wrong statement. You will tell me why it is a wrong statement. Okay, it does not comprises. It is also wrong. Gene silencing involves the methylation of H2A, H3, H4. So, although methylation is a mark which is a gene silencing mark and it is responsible for the gene silencing, but not all the methylations are involved in the repression of genes. For example, H3K4, that means in the H3 tail of the histone, at the 4th position, lysine is present. So, this kind of methylation is actually an active mark, not a repressing mark. So, that means just by saying that it is a methylation, so it would be involved in gene silencing. No, that is wrong. It depends on that which tail and which particular amino acid residues getting methylated, that is also important. So this is also wrong. The H2A H4 dimer forms a tetramer by binding to the H2B H3 dimer. The tetramer associates with another tetramer to form the core octamer. No, it is wrong. It is not the way by which the octamer formation takes place. Cytosine methylation is a well conserved mechanism for gene repression. Yes, it is true. It is a true statement. TF2H mediates phosphorylation of the tyrosine residue in the CDD of the RNA polymerase, which is required for transcription initiation. Is it TF2H? Let us see here. I think it is in lecture 5. Is it lecture 5 or lecture 6? 5. It's in 5 itself. Okay. Hmm. Correct answer is cytosine methylation is a well conserved mechanism for gene expression. Now, why the last option is incorrect? TF2H mediated phosphorylation of tyrosine residue in the CTD of the RNA polymerase 2 is required for transcription initiation. Because TFT2H is a, you can see here, sorry, I don't understand why they have not mentioned. I think H should also be correct because the phosphorylation of the carboxyl terminal domain uh, happens because of TF2H only. Can anybody tell me now? Okay. So what were your answers? D and F. So you can yourself you can see what whether your answers were correct or not. But I doubt. So eighth is the correct answer, but I doubt that seven this H should also be the correct answer. G is the correct answer. That is for sure and short. It is the correct answer. But whether H is also a correct answer or not, I doubt. I think that H should also be the correct answer. So, did you understood this question? Yes or no? Please tell me. Could you understand this question? Okay. So, basically, our topic is differential gene expression, right? But before studying about the differential gene expression, right now we are studying and dealing about how the expression actually takes place, what are the factors which are important for expression, and then eventually slowly we'll move towards the differential gene expression part. So like I told you earlier also, that for differential gene expression there are certain factors like enhancers, promoters, silencers, which are really important for controlling the expression of genes in cell, tissue, and state specific manner. Okay, now we'll come to the next question. So this was just a cartoon to this is actually important. Why? Because uh, many a times when students are asked to draw the structure of genes, they get confused, especially whether the 5 prime UTR and 3 prime UTR has to be placed after or before or inside the intronic or exonic sequences. So I would suggest you to please go through this figure. It's an important one. Okay, fine. Now next question is again a very interesting question. What will be the source of mitochondrial DNA in Dolly the sheep? Donor cell, recipient oocyte, umbilical cord of its mother, all of the other three options. So they are asking mitochondrial DNA. What is the source of mitochondrial DNA? So think and answer. Okay, interesting question. I'll give you 2 minutes for this. Don't rush to answer. Look at all the options and then think and then answer. Choose the option. Okay, now I'm switching the screen. Okay. Let us see what you have answered. Hmm. So Namanita has answered A, then B also, and Camelia has answered all of them. Okay. And Shubhamoy and Amina, what do you think would be the answer? Don't know? Think, think what should be the answer. The question is, what would be the source of mitochondrial DNA in Dolly the sheep? Donor cell, recipient oocyte, umbilical cord of its mother, or all of the other options? So, Nabanita has answered A and B, and Camellia has answered all of them. Okay. Now you see two types of DNA you can have. The one is the actual DNA which is present inside the nucleus, and the other DNA is the organelle DNA. Okay, the organelle DNA is the DNA which is present in certain organelles. For example, mitochondria. That is your organelle DNA. Okay, now this organelle, mitochondria, and you have a cell from which the nucleus is coming to this enucleated oocyte. It is a enucleated oocyte, which is nothing but a recipient oocyte, and this is your donor cell. These two options are not relevant here. So this donor cell is providing the nucleus. Only the nucleus of this donor cell comes and get here. The other thing, the cytoplasm, every other thing is not from the donor cell. The cytoplasm and everything is from the enucleated oocyte. So this mitochondria is present in the cytoplasm. That means the mitochondrial DNA, it would be from the recipient oocyte. Is it clear? Because donor cell is only and only providing nucleus. It is not providing its cytoplasm. But the cytoplasm and all other nutrients are provided by the enucleated oocyte. And therefore, mitochondrial DNA in the dolly the sheep would be from the recipient oocyte and not from the donor cell. Is this clear?
Yes, ma'am.
Okay. So that's why I was telling you it's a very interesting question, because whenever we talk about Dolly the sheep, we always say that okay, so the donor cell is providing us the nucleus. But what about the organelle DNA? We tend to forget that DNA can be present inside the nucleus, fine, but DNA can also be present in organelles. So the organelle DNA would be provided by the part which has the organelles, which contains the organelles, which is present in the cytoplasm, which is the recipient oocyte. Okay. Now next question is, oh god, again a very big question. Okay. In C. elegans, sequence X is located adjacent to the NOS1 gene. A GFP protein was fused to NOS1 to identify its expression pattern. Okay. The gene was found to be expressed in the germline. Fine. However, when sequence X was deleted, the expression of NOS1 was found in somatic cells. Based on this, which of the following statements are correct? Sequence X is a cis regulatory element required for tissue specific expression of NOS1. Sequence X is a trans regulatory element which is required for tissue specific expression of NOS1. Sequence X is a silencer that prevents the expression of NOS1 in the somatic cells. Sequence X is a promoter sequence methylated in the soma. Methyl groups may bind to H2A lysine in proteins that recognize NOS1 regulatory elements, causing its repression, and different transcription factors bind the promoter to modulate gene expression in a tissue specific manner. One, how many options? One, two, three, four, five, six options are there. Okay. Very interesting question. Give yourself time. I'm giving you 3 minutes time. Read all the options, think carefully, and then answer. It's a very good question. It will clear your concept also. So try to answer it carefully and logically. Just a minute. Just I'll again come back to the screen. Um, I don't know what happened. Anyways, okay. This is the question. God, fine. Now this is the question. Read it carefully. Think of all the possible options and then try to answer. Okay. I'll give you 3 minutes from now. It's 7:25. At 7:28, I'll switch the screen. Read all the options carefully, okay? Mm. 2 minutes more. One minute more. Okay. Now I'm switching the screen. Let us see what you have answered. So Camelia has answered E, Namanita has answered, sorry, Camelia has answered B, and Namandita has answered E. Others? Just two students. Okay, fine. Let us see now the options B and E. Okay. Let us try to solve this question. In C. elegans, sequence X is located adjacent to the NOS1 gene. So, we'll try to solve this question. This is your NOS1 gene and some sequence X is here. Okay? This is your sequence X. Now what they are saying? A GFP protein was fused to NOS1 to identify its expression pattern. So, a GFP protein here, if this is your NOS1 gene, okay? So, a GFP protein was fused with the NOS1 gene. This is what the question is saying up till now. Fine. Then the gene was found to be expressed in the germline. If this is the case, then expression is here. Expression is in the germline. Fine. Then the gene was, however, when sequence X was deleted, the expression of NOS1 was found to be in somatic cells. So, if this is deleted, then the expression is in the somatic cells. Is it clear? So, this is your gene X and this is your NOS1 gene, and NOS1 gene is fused with a fluorescent protein GFP. So when this gene X is present, expression is in the somatic cell. So when gene X is present, expression is in the germ cells. Germ cells. And when X is absent, expression is in the somatic cells. This means that this gene X is responsible for the expression of NOS1 in the germ cells. Can I conclude this from the question? Yes, I can conclude that this X protein is responsible for the expression of NOS1 in the germ cells. Why? Because when the X protein is present, then the expression of NOS1 is in the germline cells. And as soon as this X protein is deleted from here, the expression happens in the somatic cells. So now, according to this, let us see the options and try to figure out which are the correct options. So, sequence X is a cis regulatory element required for tissue specific expression of NOS1. It looks fine. Yes. Sequence X is a trans regulatory element. Sequence X is a trans regulatory element which is required for tissue specific expression of NOS1. No, because here they have said that X is located adjacent to NOS1. As X is located adjacent to NOS1, they have not said that X is located on another chromosome. That means it is not trans, but it is only cis. So this looks correct. Sequence X is a silencer that prevents the expression of NOS1 in the somatic cells. Okay, it is a silencer that prevents the expression of NOS1 in somatic cells. This might be correct. Then somatic sequence X is a promoter sequence methylated in the soma. No, wrong. Methyl groups may bind to H2A lysine that recognize NOS1 regulatory, causing its repression wrong. Different transcription factor bind to promoter to modulate expression of tissue specific manner. Okay, but not very much related to the context. So, for according to me, A looks correct and C looks correct. Let us see. Hmm. Accepted answer is sequence X is a cis regulatory element which is required for tissue specific expression of NOS1, and X is a silencer that prevents the expression. See, always remember, like I was telling you, the differential expression of gene is dependent upon certain cis or trans activating element, such as enhancers, promoters, silencers. So silencers are the elements that represses the expression of that particular gene in certain tissues or certain cells. So here you can say that X is actually a silencer which represses the expression of NOS1 in somatic cells. It only, so this is a example of differential gene expression, where you can say that because of the effect of this silencer, this NOS1 is getting differentially expressed only in the germline and not in the somatic cells, and the deletion of this X, which is a silencer actually, is causing the expression of NOS1 in somatic cells. Is it clear? So now what you have answered? Op- uh, B. Is this question clear or not? Or if there is any doubt, you can ask. If there is a doubt in any of the points. Fine. Okay. So this is what it is. Yeah. It was a big question, but it was a good question. It has, so this is how this is one of the examples that tells us that how enhancers or silencers can permit the tissue specific expression of genes. Okay. Last question. So after this question, what I have planned is that quickly, very quickly, we'll try to revise or just summarize all the important topics that we have dealt in week 2 by going through the PDFs. Okay. So this will be helpful for you to answer your assignment questions as well. So we'll try this. Now this is something that we can do in each and every class, that after discussing the questions, if all the questions are over, then we can quickly try to summarize whatever we have studied in week 2 or in that week's class by going through the PDFs. Okay, now this question is ectopic expression of which of the following factors can convert exocrine cells into endocrine cells. This is like a normal question where you have to memorize the name of the factors, but still try to answer. PDX1, NGN3, PBX1, and MAFA. I'll give you 2 minutes to answer this question. It's 7:37. At 7:39, I'll switch the screen. Okay? Answer this question. One more minute left. Okay, I'm switching the screen now. Hmm. Fine. What are your answers? Hmm. So A, B, and D, PDX and MAFA, and Ravuri Karuna Kumari has answered B. Okay. So the correct answer for this question is PDX1, NGN3, and MAFA. That means A, B, and D. So Nabandita has answered it correctly. PDX and MAFA, they are also correct. NGN3 is also the one. And Ravuri has answered B, which is NGN3. Okay, that is also correct. So the all of the correct answers options are PDX1, NGN3, and MAFA. So what are these factors? These are actually the factors which are responsible for development of beta cells. That means in the pancreatic cells, the development of the beta cells is regulated by the expression of these three factors. Okay, so PDX1 is actually responsible for the development of beta cells. It is also responsible for increasing the insulin gene transcription. So I can write it over here, or you can just, it's not that important. Just remember that these three factors are important for the beta cells expression. Your MAFA, it what it does, it binds to the enhancer or the promoter region, and it drives the gene expression in response to the glucose level. So your MAFA basically drives your gene expression in response to the glucose level, blood glucose level. And overall, the function is beta cell maturation and development. Now, what is the relation here in this context? Why we are studying here? Because the ectopic expression of PDX1, NGN3, and MAFA, it is used to reprogram various cell types into insulin producing cells. That means you can this is a particular topic that you have not studied right now. I think you will study in your week 3 class about induced pluripotent cells. So let's not get into that. Just for in a very basic way, I can tell that certain transcription factors, when they are expressed, they can reprogram cells to become some other kind of cells. Okay, reprogramming of the cells can take place. So this is what is an example here that we're discussing, that PDX1, NGN3, and MAFA, they can reprogram various cell types to produce insulin producing cells. Okay, so likewise these cells, I mean these factors, they can convert your exocrine cells into endocrine cells. So important is that these three factors, they can reprogram the exocrine cells to the endocrine cells. Okay, now let us quickly try to summarize everything that we have learned by just going through the lectures. So lecture 2 starts with the differential expression of genes. Okay, so differential expression of genes means that although same genes, differential gene expression start from the chromosomal theory of inheritance. First and foremost, we should understand that whether the inheritance is governed by chromosomes or not. So there were so many scientists that have indeed proven that yes, chromosomes are the deciding factors for inheritance, and it started from Theodore Boveri, where they showed that chromosomes are indeed important and needed for the inheritance from one generation to another generation. Then August Weismann gave the germplasm theory, which states that there is a difference between somatic cells and germ cells. That means germ cells, it can give rise to germ cells and various other somatic cells as well. Then there were other various theories which supported the germplasm theory, which has your chromosome diminution theory. So chromosome diminution theory states that after each and every mitotic stages, your chromosome was getting fragmented, but it was just a minute, I think there is, yes, Porpatham, you have raised hand, you can ask your query. If you have any query, you can ask.
Ma'am, I was registered, but this is my first class. I didn't get a notification for all other class. Can I get PPT for other classes, ma'am?
Okay. So you are not getting which PPT, like the PPT of sir, or which PPT you're talking
No,
about?
both, ma'am. First and second, I didn't get, ma'am.
Um, I mean the online classes of sir, the recorded
Yes,
sir's classes you're talking about.
yes, ma'am. Both sir and you, ma'am. I was no only introduction, ma'am. Other things I don't know, ma'am.
Okay, okay. So
I was registered for a PhD program course. This is for PhD. I need to submit certificate to my university, Chettinad Medical College.
I'm sorry, you are registered to a PhD program. That means I couldn't understand what you're talking about.
No, I was doing PhD. I have to submit with the certificate. Swayam portal. I registered this. I need to submit to the university so that I can get my credits with the certificate. So I didn't prepare. That's why I'm asking PPT to write the exam in future, ma'am.
Okay, so actually, I'm not very well aware of this system. I think it would be better if you can contact the NPTEL coordinators. But apart from
Okay.
that, I can tell that for my PPTs. So
Yes, ma'am.
on YouTube you can search Apurva Soni. I'm writing my name, Apurva Soni. Okay, so if you search Apurva Soni PMRF, so there every week whatever I'm telling in the class, so this class is actually getting recorded, and the recorded version of this class is available in your YouTube, my in my YouTube channel. So you can go and you can see from there. If you're coming in the next class, just remind me, I'll share the link also, because right now it will take time for me to get the link. So I can share the link as well. And also I think your if you are registered under this NPTEL course, I think there is a provision that you will get the slides also. So I think it would be better if you contact the NPTEL coordinators, because they would have better understanding and idea. I don't have that much idea about the technicalities, but all I can say is that my recorded classes are available on YouTube by my name, Apurva Soni. Okay. For other class, for
Thank you.
sir's
Thank you,
classes,
ma'am. Thank you.
I think for
Thank you.
Sir's, okay, Sir's classes are also available on YouTube. If you type his name, Subramanyam, I think his classes are also freely available on YouTube. But you can, it's better to contact the supervisor. I mean the coordinators of this PMRF and PTL. Okay.
And how to contact them, ma'am?
Uh, you can, if you have registered for this course, you must have some email ID. So from that email
Yes,
ID,
ma'am.
you can mail them.
Okay, ma'am. Okay, ma'am.
Hmm. Okay. Fine. So as I was explaining about the theory, that this chromosome diminution theory stated that some chromosomes are getting fragmented. So it was actually the somatic chromosomes, somatic cells chromosomes, which were getting fragmented, and not the germline chromosomes. So again there was a differentiation that there are two kinds of cells, germ cells and somatic cells, which supported the germplasm theory. Then major thing that we have studied about, that the evidence for genomic equivalence. What is genomic equivalence? Genomic equivalence means that the chromosomes are same in all the cells. That means most of the cells have the same set of chromosomes. So if most of the cells have same set of chromosomes, that means whether we are taking chromosome from an intestinal cell, or we are taking from a skin cell, or we are taking the cells from any other part of the body, it should be same. So that is what, to support this, there was a theory of genomic equivalence, and this genomic equivalence theory was supported or was evident by the uh, coming of this particular technique, which is somatic nuclear transfer technique. Okay, then again we have studied, as we have to study about the differential gene expression. Differential gene expression can be regulated at gene expression can be regulated at various levels. There can be differential gene expression, there can be selective nuclear RNA processing, or selective mRNA translation, or differential protein modifications. Then here we are talking about differential gene expression. So to study about differential gene expression, first thing is that you should understand how gene expression actually happens. So gene expression is regulated by methylation and acetylations. So methylation essentially is a mark for repression, but not always that a methylated histone will repress. No, it is not like that. You can see here that if your methyl groups binds to H3 and H4 histones, then they are actually helping in the transcription. Okay, so it is not like always in the case of histones that if there's a methylation mark then it will lead to repression. No, methylation marks on certain tails like H3 and H4, it can express also the genes. Okay, then the different parts of the of a gene. These are the different parts of a gene. It has a 3' UTR region, a 5' UTR region, a cap at the 5' end, and a polyadenylation site at the 3' end. And then we come to the various transcription factors which are required for the initiation of transcription. So when transcription is initiated in a eukaryotic cell, there is not just one or two but an array of transcription factors that sequentially binds, and it leads to the transcription of a gene. And so these are the various series of transcription factors which binds and it causes the transcription. You can read from here. And then there are various techniques which are used to study about the interaction between DNA and protein, such as gel mobility assay, DNA protection assays. And then important that we come to enhancers. Enhancer is an important topic. So enhancer is a cis regulatory element, and it controls the efficiency and the rate of transcription, and it regulates temporal and tissue specific expression. Okay, so it enhances the transcription, but it has to be within the chromosome. I mean, it has to be in the cis regulatory region. Then, to study about enhancers and to identify enhancers, there is a method which is known as enhancer trap, where we have a reporter gene and a weak promoter attached to it. We have already dealt this in detail. Okay. Then in the lecture 6, it was all about the transcription factors. This is just a table to tell you about the various transcription factors and what are their functions. Then how do transcription factors function? So basically they recruit some histone modifying enzymes and displaces the nucleosome, and this stabilizes RNA polymerase 2 on the promoter. So basically transcription factors, if transcription factors are causing transcription, so how they help in transcription? Because they are stabilizing the enzyme which is responsible for transcription. And what is that enzyme? That enzyme is RNA polymerase 2. So RNA polymerase 2 is the enzyme in eukaryotes which is responsible for transcription. So these transcription factors, when they bind to the promoter, they stabilize this enzyme. Therefore, RNA polymerase is now getting bound to the DNA region, because this enzyme is very unstable. It can loosely detach from the promoter. So, the association of the RNA polymerase to the promoter, it is stabilized by this transcription factors. Then important, again important are these are just the examples to show. Now this is important, as we also dealt transcription factor cascades. That means if there is a heterochromatin region, and this heterochromatin region, any gene which is present in this region has to be transcribed. For that, transcription factors have to come and start the transcription process. So the initial or the first transcription factor which comes and essentially starts the process, that transcription factor is known as a pioneer transcription factor. Pioneer transcription factor, and that we have studied also. That pioneer transcription factors are the one which binds to the heterochromatin region and it starts the transcription process. Okay. Then you can see here what are the pioneer transcription factor. You can see these binds to their enhancer even if they are on repressed chromatin. That means even if it is a heterochromatin, these pioneer transcription factors will come and they will bind, like MyoD, PBX. Then neural restrictive silencer element. What is this? A silencer which is causing the restriction or repression in the neurons only. That is your neural restrictive silencer element, and we have dealt a question of this also. Okay, then genomic imprinting. I will not go to genomic imprinting now because it is a very big topic. So I'll start with genomic imprinting in the next class. And I think there would be a detailed description of genomic imprinting in the next class. So in the next class, we'll be dealing. So, Porpitham. So every Tuesdays from 6 to 8 is our online class. So you can come and join there. Okay.
Okay, ma'am. Okay, ma'am.
Okay. Okay. So I think that is all for today. We have started with the differential gene expression, and we have first started with the how actually the expression takes place, and then how it is differentially regulated. We have started with understanding about enhancers and promoters and silencers, and I think this differential gene expression will be continued for your week 3 also. Yes, Monday. Namandita, Monday, but I think one Monday, which is Raksha Bandhan. I don't remember. It's 19th, I think. So I think that day your class would be shifted either to Tuesday or Wednesday. I have to see that. I have to check with the coordinators. So uh, for all other days, it is Monday only. But if there is Raksha Bandhan, then I think we need to shift it because that's a festival. So I'll see that whatever is the update that you will get by mail. So that is all for today. We are stopping the class now then.
Thank you, ma'am.
Okay, bye.
Thank you, ma'am.
Okay. Mm,